So thank you guys all for joining our fall 2021 Conservation Sciences Seminar Series. Today we have Dr. Eric Larson, and Dr. Larson is an assistant professor in the Department of Natural Resources and Environmental Sciences at the University of Illinois, Urbana-Champaign. Dr. Larson's research focuses on freshwater ecology and conservation science. Within these themes, Larson's team focuses on understanding the impacts aquatic invasive species have on their invaded ecosystems and why certain native species have declined to rarity and face extinction. Using North American native crayfish as a common model organism, Larson's research group addresses these questions using species distribution modeling, trait-based risk assessment, stable isotope analysis, and the subject of this seminar, environmental DNA. And just so you guys know, if you have any questions, you can always pop them in the chat, or once Dr. Larson is finished, you can unmute yourself and ask him directly. So with that, I will turn it over to you.
Great, thanks so much for hosting me. And I haven't developed a new department seminar from scratch in a few years now. And I was excited to kind of present on the work my lab has done in the last 5 or 6 years. And in doing that, I worry if I'm not as well calibrated to how long this should be. But the nice thing about a Zoom seminar is if I run over, you can escape, and it feels less conspicuous than in person in the room. And I'm so happy to share my lab's work with you. And my sense is that I'm presenting to a seminar series focused on conservation science. And so I'll kind of start by noting that, finding our study organisms is often that first step in conservation science. And that seems really trivial, but for some of the organisms that I work on, just knowing where something occurs and if it still occurs someplace that's part of its historic distribution is really difficult. This is a burrowing crayfish, Cambarus kazii, that doesn't live in these kind of mud chimneys in a river floodplain like you might expect of many burrowing crayfish you would find throughout the United States. It's kind of an upland burrowing crayfish that lives in heavily intermittent streams or even roadside ditches where it forms very shallow burrows, often under rocks as the stream dries. And it's a very small crayfish. This crayfish in the palm of one of my students' hand is representative of its size. Once the stream is dried and these burrows get deep, this crayfish is really difficult to find. We've got to excavate the burrows with the hope of finding a crayfish in the burrow itself, just feeling around in the dark in the burrow. And there's many burrowing crayfish in kind of southern states of the US. Just because you find a burrow doesn't mean that the crayfish in it is this kind of rare animal that we're looking for. So we had a recent project with a master's student, Kathleen Kibido, here with a quadrat, where the state of Arkansas is concerned. This is a species that may end up being a candidate species for listing under the Endangered Species Act. And the last time someone looked for this organism a few decades ago, they just couldn't find it. So we had a project to try to find Cambarus kazii, where we went out on kind of stratified random sampling across the presumed historic range of this organism, set out random quadrats, looked for burrows, tried to excavate these burrows. Sampling for burrowing crayfish is extremely hard. It's hard to dig these burrows out. You're destroying the habitat for this organism that after you've collected it, you're going to put it back in the burrow, pad the dirt back in and hope that it's okay. And, like past conventional sampling for this animal, we just didn't do that well. And there's lots of similar just crayfish among other organisms that are very hard to assess kind of population status and trends to know if they're doing okay. So there's been a big boom and interest in kind of alternative methods for these types of difficult to detect organisms. And that includes approaches like citizen or community science, where potentially birders or users of a phone app like iNaturalist might be able to report records of interest to a management agency, interest in remote sensing, which can include a lot of different techniques to observe the environment. And a big boom in molecular surveillance that has grown from things like maybe kind of passive hair traps to do population genetics on mammals to these ideas, these approaches around environmental DNA of collecting environmental samples, soil or water or air, and being able to identify the DNA of organisms that have been in that environment, hopefully sometime recently. So we actually recently tried environmental DNA as a complement to our conventional sampling for this upland burrowing crayfish in Arkansas. And I'd say our conventional sampling across 2 springs, 2 wet seasons, we detected this animal at 9 of 51 sites. These 51 sites were chosen from a kind of Maxent species distribution model to be good places we expected to find this animal, and even starting from what we assumed was good habitat for it. It's just hard to find. When we compared that to environmental DNA, taking water samples from these same intermittent streams when they're flowing in the spring, we were only able to sample in 1 spring at 25 of the sites, a subset of half of our same sites from the conventional sampling. That is thanks to COVID-19, as many of us probably struggled with trying to do field biology in the last 2 years. But we detected this animal at 6 of 25 of these sites, 24% to 17%. Neither of those values is exactly a home run, but eDNA is a lot easier than digging up burrows, and we're not destroying the crayfish's habitat. And once we've developed an eDNA assay, this is pretty cost effective relative to the kind of personnel time and hours to do our conventional sampling. So we're in the process of writing up our final report for the funder for the Arkansas Game and Fish Commission, where my recommendation would really be the future of surveillance for this organism should be environmental DNA. And this is a direction that a lot of monitoring for invasive or threatened and endangered species is trending. And so I think that's why I'm here today, is to kind of talk about my lab's experience. One of probably hundreds in the United States has worked on environmental DNA issues in the last decade, to kind of talk about our experiences and lessons from eDNA with a focus on kind of the sampling design and field biology side of environmental DNA. And as I mentioned on a previous slide, there's kind of contentious arguments among the eDNA community about definitions and what is or isn't eDNA. I often think of this approach, environmental DNA, as DNA that we've captured and identified from an environmental sample. And I'm an aquatic ecologist. I often think that's a water sample, but that can be soil, and in some cases that could be air. And this is not a new idea. This is something that microbiologists or microbial ecologists were doing for a long time before people like me, a fisheries biologist or conservation biologist, considered applying for macrobiota. And generally, when we're looking for something like a crayfish or a turtle or a fish, those macrobiota, our environmental DNA, is generally something we've collected without collecting the target organism itself. These are our sloughed cells or extracellular DNA. And there's kind of a family of techniques under this eDNA umbrella. Some of these techniques focus on single species or target taxon. We've done eDNA at kind of a genus rather than a species level in some cases. That often uses approaches like quantitative or real-time PCR that's very similar to the type of COVID-19 test you might take for viral surveillance. You can also apply eDNA to whole communities by kind of high throughput or next generation metabarcoding approaches. Almost all of my work and my lab's work has been single species qPCR work, but I think some of the things we've learned about how the environment and organisms affect eDNA's detectability to our sampling is going to apply in these kind of whole community metabarcoding contexts as well. And this is something, as I mentioned, has really boomed in popularity as it's grown from kind of a microbial ecology tool to applications in macrobiota. I think a lot of people would attribute this as really picking up attention and emphasis from a 2008 paper out of France that was using environmental DNA samples to look for invasive bullfrog. Our native bullfrog is a non-native organism in Europe. sampling ponds that had had conventional surveys for bullfrog and comparing water samples that had PCR run from those water samples and finding really good performance. You could tell if there had been a bullfrog in these farm ponds in France just from a water sample. In North America, published a few years after this, but kind of developing concurrently, eDNA really grew out of interest in improving surveillance for a number of invasive species like silver and bighead carp as a risk to spread from the Mississippi River Basin into the Great Lakes. This idea of trying to have invasive species surveillance approaches that are very sensitive to the presence of organism at low abundances when a management intervention or an eradication effort might actually be effective. And this is kind of a fine line to walk, right, of wanting invasive species surveillance that's really sensitive to the earliest possible warning that organism's there, but that also maybe doesn't give you false positives that result in management responses when the organism isn't there. But this idea really rose to prominence around silver and bighead carp surveillance that's moved to kind of federal and provincial level surveillance for invasive species for these fish in the Great Lakes Basin in the US and Canada. So if we think about that context, and I know this is such a cliche to show a keyword search from Web of Knowledge, but here's the topic eDNA in a kind of database search, limited to the year 2008 or later after our kind of bullfrog paper from France, and restricting this to just a few Web of Science categories: ecology, biodiversity conservation, marine and freshwater biology, fisheries, zoology, limnology, trying to exclude microbiology papers from this literature. We have gone from there being basically no eDNA papers or papers authored by someone named Edna, as my earlier searches were returning, almost no papers in 2008 to 2009, to 200 papers published in 2020, and we've got a quarter of a year left to go in 2021. There'll probably be 250 to 280 eDNA papers for macrobiota published this year. Citations per year of these papers in a range of about 4,500 at present. And this is a lot. When I tallied this up, this is 890 publications since 2008. If you're a graduate student interested in eDNA for thesis or dissertation work, this is something like species distribution modeling in the early 2000s, doing this exponential growth thing where you maybe feel a little overwhelmed. How do I possibly keep up on this literature that's growing by maybe a paper per day in 2022? This is a lot, but we might ask, in 890 papers, we should be starting to know a lot about this, right? And I would say a lot of that work, from my perspective, has had a lot of emphasis on the capture and preservation and extraction and quantification of eDNA. A lot of it is very focused on eDNA maybe as a technology and less on the sampling design or the field biology of eDNA. But I think there are a lot of kind of generalities and moves towards consensus on this side that I think are really valuable to be aware of. And I think this eDNA research community from Europe to North America to Japan is moving steadily towards consensus on how to design and validate these single species type qPCR assays, what qualifies as an eDNA detection, and how you can quantify eDNA concentration in these type of samples, moving more towards some standardization of how to collect DNA in the field and extract it in the lab, increasing consensus that eDNA does correspond well with abundance or biomass of target organisms, at least under controlled or experimental conditions. And in some ways that breaks down in field systems, and we'll kind of look at some examples of why. I think we're less far along on standardization towards eDNA metabarcoding of whole communities. It has a lot of decisions you've got to make around bioinformatics pipelines that I think we're still doing a lot of work on how best to apply eDNA in those cases. But briefly, I just want to run through some of these papers that I think, as we've done eDNA research for over a decade, 890 papers, It's nice that we're starting to get some synthesis, some meta-analyses, some review papers that can give you relatively good guidance if your lab is thinking about getting into eDNA on where to start for kind of recommended practices from assay design through lab procedures. So one of these that came out, there is now a Wiley journal, Environmental DNA, which is a very useful journal that is picking up a lot of these review and meta-analyses papers in eDNA. One of these is a validation scale to determine the readiness of environmental DNA assays for routine species monitoring. Let's say you're looking for a species like our Boston Mountains crayfish, Cambarus causii. You want to build a new single species qPCR assay for that crayfish. This is kind of a guideline of the steps for that assay to be generally acceptable to kind of the scientific community in eDNA work. And I think we're moving towards agreement on what a good single species eDNA assay looks like. Similarly, published recently in that same journal, Environmental DNA, we're approaching some agreement on what counts as a detection from an eDNA sample, and when do you have an eDNA assay that you can confidently quantify eDNA concentration or copy number out of? How do we calculate a limit of detection and a limit of quantification where labs in different countries or regions agree about what those terms mean, what a limit of detection is, what a limit of quantification is for these type of assays? A really nice review paper from a few years ago, Critical Considerations for Application of Environmental DNA Methods to Detect Aquatic Species. This is led by Karen Goldberg at Washington State, who has a very productive eDNA lab that does a nice job of walking through step by step where there are pitfalls or risks of starting an eDNA program in your lab. I think one thing as a reviewer or a journal editor that I occasionally bump into is molecular labs. We've done qPCR. We're interested in getting into eDNA. One recommendation that a lot of people in this field feel very strongly about is needing separate clean room handling spaces for eDNA samples relative to where, say, high copy DNA is being produced by PCR or qPCR machines. That physical separation between sample extraction and PCR is something that a lot of people doing eDNA work very much look for example, at peer review as you try to publish an eDNA study. The journal Environmental DNA also had a review of methods for collection, extraction, and detection of eDNA samples. This is just kind of a census of what people are doing to capture and extract eDNA in the last decade of work. They've got a review here with 369 papers that met their criteria. And they found, at least for aquatic studies, most studies were capturing DNA by filtration rather than other methods. The most common filters are cellulose nitrate filters, which a number of method studies do support as performing well. And there's a number of kind of standard pore sizes for those filters that a decent part of the community are using. I think there's some interesting work to go on relationships between filter pore size and eDNA relationship to biomass or abundance. I've seen some papers in review as a journal reviewer that have been arguing that larger pore sizes that capture more intracellular rather than extracellular DNA may produce better relationships between organism biomass and eDNA copy number than really fine pore sizes. Capturing all DNA might weaken our relationships to biomass relative to capturing just the most recent DNA, which I think is an interesting idea. Of this review, most of these eDNA studies, over 75%, are using a commercial DNA extraction kit to extract their DNA. And the most common of those kits is a QIAGEN DNeasy blood and tissue kit. There may be better ways to extract DNA from environmental samples. My lab and my collaborators at the University of Illinois really like to store our samples in buffers like Longmire's or CTAB and do a phenylchloroform isoamyl approach for extraction that we think gives us higher DNA yields with fewer PCR inhibitors. Chemicals can stop our qPCR process from working as well than using these kits. But I think there's probably some benefits over time to standardization between labs. I might argue for a conventional sampling technique. I don't know why your backpack electrofishing. I think an electric seine is better, but if multiple state wildlife agencies agree on and standardize a technique, we may have better prospects of transferability of data between universities or agencies or people monitoring the environment. And I think we've kind of moved incrementally towards some of the standardization between labs over time. And how well does eDNA do? In my experience, eDNA often performs very well for occupancy, for presence, absence of an organism. I mostly do field rather than laboratory studies, and we've felt some pessimism about the ability of eDNA to represent abundance or biomass under field conditions, an actual lake, an actual river. But we're starting to get enough studies asking that question. Does eDNA copy number or concentration from a sample correspond to biomass or abundance of the organism you're looking for? This is a meta-analysis that came out a few years ago that looked at these correlation coefficients and R-squared values from both laboratory and field studies between eDNA concentration and the abundance or biomass of organisms. And they found that eDNA is strongly related to abundance or biomass under lab conditions. You've got a R-square across the, I think 30 or 40 lab studies that were included in their meta-analysis of about 0.8, but that drops off between 0.5 and 0.6 for those field studies trying to relate abundance to eDNA concentration or copy number. So we have a much noisier relationship under field conditions in the performance of eDNA. And why would that happen? That's the abiotic environment being noisier and less controlled than this lab mesocosm. That's potentially the biology of that organism. Is it in the life history of the fish you're looking for, did you catch a spawning event that's resulted in a lot of DNA in the environment that might even decouple from that kind of adult biomass you're trying to relate to? I would also say this may be noisy because when we're doing field studies, our conventional sampling methods also have error, right? In our lab aquarium or mesocosm studies, you've stocked a known density of tadpoles in a cattle tank, and you've got a pretty confident biomass number you're going to relate eDNA concentration to. When we're doing this in the field, that's trapping crayfish or setting hoop nets for fish, or doing snorkel surveys for hellbender salamanders. Some of this error isn't necessarily on the side of eDNA, but on the side of our conventional sampling for density or abundance or biomass. If we kind of look at this increasing synthesis of eDNA work over the past decade, it feels optimistic relative to where we were in 2015 or 2016 when I started doing this. The people who may peer review your paper or want to use your eDNA assay at a management agency increasingly agree on what a good qPCR single species assay looks like, what a detection should qualify as, what's a limit of detection under a qPCR run. Are you counting something aberrant that happens at cycle 50 on your PCR machine, or have you run a serial dilution of some standard to know reliably what concentrations you can detect across samples? I think there's always that kind of impulse to build a better mousetrap, but I think we'll eventually have to agree on standardizing collection and extraction, and the benefits of sharing methods between agencies or labs may outweigh the kind of search for the best collection or extraction approach. And we're increasingly confident that the amount of DNA we find from an environment sample has some relationship to the abundance or biomass of the organisms we're looking for. It's just that relationship gets muddy when we're in that real kind of field condition that we care about. So if we say across this 890 papers, we've made this progress, what's left to do? What do we need? And I kind of return to this issue in this meta-analysis of our relationships between eDNA's performance and what we know about the organisms we're looking for, breaking down more under field conditions. That feels awfully important. no one's doing this to know how many tadpoles are in your mesocosm. We're doing this to know, are there Boston Mountains crayfish in this intermittent stream or not? That variable, hard to sample, real environment where we need some help knowing something about the population status of a really rare organism. So this is where the title for my talk comes from, is this idea of the ecology of environmental DNA, how the origin and fate and transport of DNA in the environment is so strongly influenced by the life history of the animals we're looking for and the timing of environmental conditions. And this isn't an idea that starts for me. The co-authors here, Matt Barnes and Cameron Turner, were PhD students at the University of Notre Dame, where I was doing a postdoc at the time, and they wrote this really nice review paper about all of the ways that ecology interfaces with eDNA rather than necessarily our lab-based technology side of how eDNA should work. So that is that the origin of DNA in the environment may time to the reproduction or mortality events of the organism we're looking for. How much DNA is available in an environmental sample varies by degrees of magnitude over time in relation to the life history of the organisms we're looking for. How long DNA persists may depend on environmental conditions like temperature or UV exposure or the biomass of the organisms we're looking for. And things like transport. If we're in a river and DNA is trapped in sediments, potentially after an organism no longer occurs at a site, as a flood mobilizes DNA out of those sediments, you could experience false positives from transport in the absence of an organism being there. The kind of leading edge of research that we need to solve is how our organismal life history and environmental conditions are influencing the performance of this approach. And so my lab has tried to work more on that field or sampling design piece of eDNA rather than kind of the lab bench, how do we best remove inhibitors kind of side of eDNA. And I would just really stress, eDNA is not a silver bullet that's going to work identically across every context, regardless of the biology of the organism you're looking for or environmental conditions. And I think there's sometimes a misconception that whether the animal you're looking for is abundant or rare, eDNA should always find it. I would say instead that you still really need to think like a field biologist in designing sampling schemes that increase the likelihood of encountering that eDNA that you're looking for when your organism is actually present. And I think there's probably limits to the ability of better lab methods to resolve things like underpowered field sampling, not taking enough, let's say, water samples or high enough volume water samples, or samples collected at the wrong time of the year when your organism isn't active or isn't reproducing and is really tough to detect. So I think we're getting better about this, and more labs are kind of adopting techniques from fields like conservation biology or wildlife management. Something that I like about environmental DNA is the replication of environmental samples in the field, and often PCR subsamples on a plate are technical replicates being pipetted into a plate before it goes in a qPCR machine. Gives us a replication that lets us model things like occupancy and detection probability and these kind of hierarchical frameworks that are very common or very popular in the wildlife biology literature, right? So we may have a two-level model where we're interested in occupancy probability, whether the organism, or in our case, its DNA, is actually at a location or not. But we're also interested in detection probability. Do we detect that DNA when it's there? And we can use these replicated, either plate replicates or field replicates, these zeros and ones, to estimate a detection probability that is often driven by a different process than occupancy, than the presence or absence of your organism. So it's kind of nice in an eDNA study, we may think that our occupancy probability might be driven by the organism actually being at the location. We are modeling occupancy for DNA. But if we have conventional sampling data as we're maybe testing a new assay, the abundance or density or biomass of our target organism could be a predictor for occupancy of that DNA. We found the Boston Mountains crayfish at this site. that should be a good predictor that the site is, has DNA there. But detection probability might be affected for DNA by a number of different environmental factors than necessarily affect the occupancy of the organism itself. How long does DNA persist at your sample location? And that might relate to stream discharge, transporting DNA downstream, or UV exposure or temperature. And so we can end up with different covariates for occupancy and detection probability that can work with this variance in detections or non-detections in our field replicates. So my lab has tended to apply these occupancy modeling approaches in single species qPCR assays to investigate how the ecology or the life history of organisms and environmental variability affects our ability to detect eDNA. We have done this both for threatened and endangered species as well as for invasive species. And I'll just kind of walk through a series of short methods-light examples of the kind of types of life history or environmental conditions that we think are going to affect eDNA's performance, independent necessarily, I would say, of better lab methods. So the first of these is kind of focused on how organism behavior or life history may affect eDNA's performance. This work was done with Leslie D'Souza, who was a postdoc working with me when I started my lab at the University of Illinois. Leslie is now in the conservation science team at the Field Museum in Chicago, and Leslie's very amazing. This is a project Leslie brought from past work in Alabama with the Alabama Natural Heritage Commission, Jim Godwin, where Leslie and Jim were trying to use eDNA to look for a rare turtle, the flattened musk turtle, which is an ESA-listed species, and an obligate aquatic salamander, the black warrior water dog, which was a candidate for listing under the Endangered Species Act. After our study, the black warrior water dog was listed as ESA threatened. To my knowledge, this was the first case where an eDNA study was used in an ESA listing decision. And Leslie and Jim had sampled across multiple years, across many seasons, looking for these species in Alabama. And something that I thought was interesting as Leslie had this data set they were trying to write up is that the flattened musk turtle is warm season active. It warms up and this turtle's out, in July, in August. You'd be more successful trapping for this turtle in the summer than you would in the winter. And the inverse is true for the black warrior water dog that is cold season active and reproduces in the winter. eDNA is kind of nice because the same water samples that have been filtered and had DNA extracted out of them can be reused to look for multiple species. So we have the same samples with two different single species eDNA assays for organisms that just by the timing of their activity, we expect might differ in their detectability by eDNA. So here's a few maps. We have non-detections for each species in orange and detections in red. I made these slides on a much smaller laptop, and only when I blew them up on my work computer this morning did I realize they're fairly low resolution images. Sorry about that. A decent sampling extent. Both species largely persist in the protected areas of national forest lands in the Locust Fork in Alabama, largely extirpated from much of their historic range. Again, the musk turtle, warm season active, black warrior water dog, cold season active. We ran occupancy models with AIC model competition. I am showing a power analysis of the outcome of our best models. How many samples do you need to detect the flattened musk turtle or the black warrior water dog when they're actually present with a 95% confidence of detecting the species? In all of our occupancy models, season, warm or cool season, was included as a well-supported predictor for detection probability. On the top here, we have warm season samples needed to detect our flattened musk turtle. The warm season samples are in red, the cool season samples are in blue. We've got a dashed line for 95% detection probability. If it is the summer and the flattened musk turtle is actually at your site, you need 3 to 4 1-liter water samples to have a 95% probability of detecting the species. If it's the winter, if it's the cool season, you need something more like 12 to 15 samples to have the same probability of detecting this. And the inverse is true for a black warrior water dog. And this model's super messy because we don't detect that many black warrior water dogs. But the thing that matters to me is that effective season on detection probability inverts. This species is more detectable in the cool season. It's less detectable in the summer. We would estimate with fairly high error here that you might need around 10 samples in the winter to have a 95% detection probability for the species. You're going to need a ton of samples, 30 or 40 in the summer, to detect this organism that's not active in the warm season. And you can imagine if you're the Alabama Natural Heritage Commission and you're trying to use the same water samples to monitor for multiple threatened and endangered species simultaneously, this is potentially inconvenient, that the optimal time of the year to sample for multiple co-occurring species isn't the same time of the year, right? Your solution to this is probably, if our black warrior water dog needs 10 samples in the winter, and that gets you to a 95% detection probability for the flattened musk turtle, 10 cool season samples is probably better than 40 warm season samples to get your black warrior water dog. But I share this interest in kind of trying to optimize sampling for multiple species simultaneously with Gretchen Hansen at the University of Minnesota and Josh Dumke and Chanlan Chun at the University of Minnesota at Duluth, where we're trying to do this on a collaborative project right now. If the availability of eDNA to detection varies in time in different ways for different species, how do you kind of solve an optimal time and cost-effective sampling regime? So that's this kind of organism or life history piece, same samples taken over multiple years and multiple seasons. We expect that eDNA is affected by environmental factors. This is a study, again, with a turtle. This was a study we did a few years ago. Here's Ethan Kessler, our lead author. He was a PhD student at the time. He's now a postdoc with the Illinois Natural History Survey. Alligator snapping turtles have been a species that was petitioned for listing under the Endangered Species Act. Alligator snapping turtles were entirely extirpated from Illinois, probably by over-exploitation. The Illinois Department of Natural Resources and several local zoos kind of started a captive breeding and Head Start program to reintroduce alligator snapping turtles into Illinois. This has been going on for several years. They've been reintroduced into Clear Creek, a stream in southern Illinois on the Missouri border. And the turtles that are reintroduced are tracked by radio telemetry. And we thought this was a really exciting opportunity to investigate the performance of eDNA in a system that is a natural system under field conditions, but has a lot of the kind of control of a laboratory or mesocosm experiment. We think we know where every single alligator snapping turtle in the entire state of Illinois is. They all are being tracked by radio telemetry. So we could go out and take water samples directly on turtle locations and downstream of turtle locations and do occupancy and detection probability modeling related, for example, to upstream turtle biomass from our radio telemetry tracking. And that's what the map here is showing. Our turtle locations are in white, random locations, downstream locations are in black. Larger circles, we had more positive detections per field replicate, and smaller circles, we had fewer. Those kind of variance in detectability that we're feeding into our occupancy models. Beyond relating eDNA to radio tracked animals in a reintroduction, something that Ethan did that I thought was really clever was he used remote sensing data to delineate how much of the upstream river channel was open to UV exposure as opposed to closed by the riparian forest canopy. And there have been some laboratory and mesocosm work on the effect of UV exposure on DNA persistence time and detectability. But this was a really nice covariate to be able to include as turtle biomass, hopefully affecting occupancy of DNA in our samples, but UV exposure, channel openness from remote sensing as something that might affect detection probability, hopefully without simultaneously affecting where our turtles are choosing to be. Maybe that's not true. Maybe a nicer riparian forest has more of that large wood where our alligator snapping turtles want to hang out and ambush fish. And I thought this was a nice mix of radio telemetry, remote sensing, and eDNA to evaluate eDNA's performance. So in most of these cases, we're running models that we compare by AIC in a kind of information theoretic model competition approach. And what Ethan found was that our DNA occupancy increased with upstream turtle biomass, but DNA detection probability decreased with increasing openness of the canopy, increasing UV exposure. And this is what this looks like. Like a lot of field biology, this is noisy. We've got R-squareds here of about 0.38 in our most supported model. As our alligator snapping turtle biomass upstream of the sample increases, and Ethan tried a series of buffers, 100 meters, 500 meters, 1,000 meters. 1,000 meters was our best buffer for upstream biomass. As our alligator snapping turtle biomass increases upstream, this is grams of turtle, eDNA occupancy probability increases. We hit 3,000 grams of alligator snapping turtle somewhere within a kilometer upstream. You've got a pretty good chance of having a positive eDNA detection. But we also have detection probability decreasing with that canopy openness. The more the stream is exposed, the less eDNA is sticking around, we believe, due to UV exposure. This was a stronger relationship from a different model that looked at eDNA concentration, copies per microliter. eDNA copy number declined precipitously with canopy openness increasing. This is what's driving that relationship to eDNA detection probability. The amount of eDNA in our samples, the concentration is decreasing as there's more sun exposure to our channel upstream of where the sample was taken. I've also been curious. I work in lakes, but a lot of my training has been as a stream ecologist. There's been interest among multiple research teams to investigate how stream flow or discharge should affect the performance of eDNA. Should higher stream flows dilute eDNA, or should higher stream flows potentially increase detections by transporting eDNA farther? And this is something we chose to investigate locally around the University of Illinois campus here using non-native corbicula clams. We chose corbicula as a species, as a taxon that we had a good genus level eDNA assay for. Corbicula clams are very common. They're very abundant in our very agricultural streams in central Illinois. And so we could go out and sample through the year at stream sites with USGS flow gauges that would allow us to relate discharge to eDNA detectability and concentration for our corbicula clams. This is led by Amanda Curtis, a PhD student in my lab here. Amanda and I are at one of our higher flow days at one of our sites with a USGS flow gauge. You can see the USGS gauge over Amanda's shoulder on the opposite bank on the bridge here on the Salt Fork of the Vermilion River in central Illinois. We had 2 stream sites that we sampled continuously through 1 calendar year, January through December. Salt Fork and Copper Slough, these are 2 of the USGS flow gauges closest to us here in Urbana-Champaign. You've got the hydrograph here from those flow gauges in blue. You have temperature through the year as black diamonds at the spot sampling when we were out at the stream sites, from very low temperatures in January to kind of typical stream temperatures around 25 Celsius in the summer. And our drop lines in gray are the sampling events we had both at Salt Fork and Copper Slough. We visited each stream several dozen times through the year, both at base flow and high flow conditions. We had a series of 6 other sites that we visited in the summer and the fall at low and high flow conditions. And we sampled all of these sites in the summer with a conventional quadrat approach for their corbicula clam densities. So why do this? When we've talked about this work with people, that the reaction is often, well, it feels obvious that the higher discharge should dilute eDNA and make eDNA harder to detect. But our thinking was that there were several potentially opposing processes that could make a study of stream flow effects on eDNA in the field worthwhile. As stream flow increases, we do expect that DNA should get harder to detect. It should dilute. Here's the Salt Fork at that same site from a summer base flow to kind of an early spring flood. It feels like corbicula DNA should be harder to find in the larger version of this river. This could also make DNA harder to find at high stream flows if we're experiencing PCR inhibition. If tannins or humic acids or other PCR inhibitors have been mobilized from the sediments by the floodwaters. Maybe our PCR process isn't working as well. That's something we can check and improve on the lab side, but it's a reason a flood or a high discharge event could make DNA harder to find in the environment. But on the flip side, there's been relatively good work that DNA is transported farther under higher discharge conditions. In a small first order stream, we often expect that DNA is only transported 50 meters to 100 meters. In a fifth or sixth or seventh order river with a higher flow volume, we often find that DNA can be transported several kilometers or tens of kilometers. So if we change discharge in one river, maybe we're just increasing the river length that we're detecting corbicula eDNA from. We also have potentially corbicula DNA, older DNA, not ancient, but old DNA trapped in our sediments that's been mobilized with the flood, right? Maybe we're getting DNA mobilized out of sediment rather than DNA that's being detected from our live corbicula nearby in the stream channel. So we have these opposing mechanisms at high stream flow events, and we wanted to know, what does this translate into in an actual invasive species surveillance program? How bad would it be if you're doing your eDNA sampling for a high priority invasive species, and it's on your work week, there was a big rain event, this is the only time you're going to be at this site for this entire year, and it happens to be flooding, you need to take the sample. Did it matter that you sampled on a high flood event because that's when you were in the field? Something that could very plausibly happen to a management agency biologist. Finishing your rotation on the week, you've got a flood, that's when you could sample. So a few things that dropped out to us, like our flattened musk turtle or black warrior water dog work, we get a strong season effect on corbicula eDNA. These are our concentrations, these are our copy numbers. Corbicula eDNA increases dramatically with temperature. As it gets warmer, corbicula eDNA increases by several degrees of magnitude. Corbicula reproduce around 20 to 22 degrees Celsius. Once you hit May or June and these streams warmed up, the amount of corbicula eDNA samples really boomed. Good to know, if you were looking for corbicula, you've got a pretty good target in terms of time of the year or water temperature to look for the species. We're able to control for this simultaneously in our models, though, where we find that eDNA concentration also decreases with increasing stream flow. And this was included in all of our most supported models for corbicula eDNA occupancy and detection probability. These are concentrations, but also for our occupancy models as well. Copy numbers of corbicula DNA are declining with increasing stream flow at these sites. We're seeing dilution winning out over maybe mobilizing or transporting corbicula eDNA from farther. On the occupancy side, here's temperature and stream flow at our 8 sites that we sampled both in the summer and in the autumn. We related corbicula density from our conventional sampling to copy numbers. Here's that positive but noisy relationship where eDNA does okay, but not very precise of a job of relating to abundance under field conditions. As corbicula density increases from no corbicula to 112 per quadrat, copy number increases. But these models had strong effects of season between summer or autumn. And this is that power analysis idea again. This is looking at the number of samples you would need to have a 95% probability of detecting corbicula in the autumn versus in the summer on a gradient of corbicula density on the x-axis. Corbicula is very easy to detect even at low abundances in the summer. Go out at something like a low flow or base flow event in May or June. Even where there's very few corbicula, you're going to be able to detect that species with 3 or 4 water samples. But as you move into high flow, low water temperature conditions, maybe in November, even at densities of 60 or 100 corbicula per quadrat, you may not get detections. So a lot of my lab's work just wants to stress that methodology in eDNA capture, storage, extraction, quantification may matter less going forward to our performance than how eDNA is produced and transported and its fate due to the field biology side of the ecology of eDNA. And I know we're at 12:50. I want to leave a little time for questions. So I do just want to touch on a few cautionary tales from our lab, a few quick snapshots of eDNA not always being your best approach, and these kind of risks of the file drawer problem that I sometimes think our eDNA literature doesn't always represent the pitfalls of the methodology very well. Very briefly, this is an example of another ESA candidate endemic crayfish. This is in the Missouri Ozarks, the cold water crayfish. This has a very good conventional sampling approach. Put down a meter squared quadrat, hold a seine below it, kick over the rocks, and you catch crayfish. And the only thing this costs you is occasionally your dignity, depending on your form for your crayfish shuffle. We wanted to compare our kind of quadrat seining approach to eDNA for this main stem kind of large river but rare crayfish. And I would say they did pretty similarly, but conventional sampling detected this rare crayfish at 2 sites where eDNA did not, and eDNA did not detect this crayfish at any sites where the conventional sampling failed to detect it. So if you look at this map, we've got some black X's that are sites that dried. We didn't sample those because this crayfish doesn't tolerate intermittent streams. We have some purple circles. Those we sampled with both techniques. We didn't find our big main stem crayfish there. We have some yellow circles. Those are places where we got the crayfish by conventional sampling and eDNA. And then we have some green circles on our upstream range limit of this rare species. Those are places where we only detected this organism by conventional sampling. We didn't detect it by eDNA. When we did some occupancy modeling in the system, we found a really strong effect basically of river mile or downstream distance on eDNA detection probability. The farther downriver you went, the more eDNA there was in our samples, the more likely you were to detect the DNA of the species. And this actually decoupled in the river from where the crayfish was most abundant by our conventional sampling. We've got a map here of density as these white circles and our detection probability in red. Detection probability increased as our DNA was being transported downstream from the best habitat in the species range. This is a groundwater fed, cold, swift flowing river where I think our DNA is being well preserved as it's transported downstream. DNA is very detectable outside of kind of the optimal habitat for this species, but the species is basically undetectable by eDNA at its uppermost range extent where we only found it by conventional sampling. There is no good argument in this case to use eDNA for this organism, given how easy and non-destructive our kind of seining approach is. If you wanted to repurpose an eDNA sample to look for this crayfish, it would work in a lot of contexts, but it shouldn't necessarily be your first approach for this animal. I also very briefly want to talk about one that is in press. This should be out in a week or two. A study where we were trying to develop an environmental DNA assay for the virile crayfish, a non-native species in the Ozark National Scenic Riverways, a national park in the Missouri Ozarks. As part of this good practice of building a single species qPCR assay, we wanted to go out and sequence the mitochondrial DNA of all of the native crayfish in that watershed so we could build an assay that only amplifies the DNA of our non-native virile crayfish that's specific to virile crayfish. And as we did this, we kept bumping into native crayfish that had virile crayfish mitochondria and virile crayfish that had native crayfish mitochondria. So we followed up with some sequencing in the nuclear genome, and we found that many of our non-native virile crayfish, they look like virile crayfish by phenotype, they have a virile crayfish nuclear genome, had our native spot-handed crayfish mitochondrial DNA. They had hybridized. And the kind of direction of hybridization in the system, it was much more likely that the non-native virile crayfish had native crayfish mitochondria than the inverse. We've seen that in other invasive crayfish hybridization systems. Something else that happened that we've seen with hybrid fish in the West, hybrid trout, it may be that our hybrid individuals are more likely to disperse than our non-hybrid virile crayfish non-natives. So our hybrids are shown in this map of the current river in Missouri as a lighter shade. Red are virile crayfish that have virile crayfish mitochondria. Lighter colors towards white are virile crayfish that have native crayfish mitochondria. On the leading downstream edge of this invasion, something like 40 or 50 river kilometers, all of the virile crayfish we found were hybrids. They all had native spot-handed crayfish mitochondrial DNA. Our environmental DNA assays work on mitochondrial DNA. not impossible to do nuclear DNA, but it'd be very hard for crayfish for an eDNA application. So our environmental DNA assay could not detect these viral crayfish hybrids at the leading edge of their range expansion. And in general, through this whole watershed, our eDNA assay performed very poorly for viral crayfish. And I just want to, briefly say, I am an eDNA optimist. I was a skeptic when I first started doing this work as a field biologist in 2014, but I do have some perception that the file drawer problem, the tendency for negative results to not get studied, can result in our eDNA literature being overly optimistic about how likely the approach is to work. Negative results are hard to publish. If you developed a single species assay and compared that to conventional sampling and eDNA didn't work, you will be put through a ringer by reviewers of everything they think you did wrong in the lab and field. And even if you very carefully documented your lab and field methodology, something that you would say, hey, we didn't get detections in this case, don't repeat this approach then for this species, we still find it very daunting to publish a negative result in the eDNA literature. So my lab has multiple unpublished eDNA studies where eDNA didn't work for us. I talked to colleagues that have had the same experience, but for my lab, we happily publish the 2/3 of cases where eDNA works and find ourselves deflected by peer review in the 1/3 of cases that don't work. I have some worries that we have a literature that does have some file drawer bias about how well eDNA should work. And I just kind of want to say, if eDNA isn't working for you, you may not be wrong, and the cause may not be your lab technique, but it may be the field biology or natural history of your organism. And I know I've run long. There are a lot of people to acknowledge, but I would really say this work was almost entirely 90% funded by USDA hatch projects, cases where conventional studies for something like the Boston Mountains crayfish, we were able to amend a little eDNA work with USDA hatch funds. And that's made a lot of this work in my lab possible. And I'm happy to stick around as long as people are patient and willing to stick around and ask questions. And we have one in the box, what's the average threshold concentration level of DNA in an analysis? This kind of idea of limit of detection, the approach people are moving towards is taking a standard that might be a synthetic DNA, maybe a synthetic G block, or genomic DNA that's been run out on a serial dilution where you've quantified that concentration, and defining the limit of detection as that concentration. People will disagree, but often the concentration where, say, 1 out of 3 wells on the plate reliably amplifies on that standard. And that concentration varies from assay to assay. And I don't know that someone has done a meta-analysis on what's the average limit of detection. I think it's a little affected by your PCR settings and the assay. A question about eDNA for rare tree detection, red mulberry, hybridize. Yeah, the hybridization question. There's both hybridization as a challenge. I also think we have had cases where for closely related species, we have struggled to develop for animals mitochondrial DNA assays that can really differentiate maybe one non-native species from a closely related native congener. That was our case trying to develop eDNA for alewife, where we weren't satisfied we could get an assay that would only amplify alewife eDNA and not closely related, other kind of river herrings. You can get a sense of that. There has been a lot of movement towards kind of in silico computer, primer design and testing work. You can do a lot of your assay design on a desktop before you've started a study to try to figure out if your assay should work for your species, or if you're unlikely to be able to differentiate a target species from a closely related non-target. Nick asks, how do you think community scientists could be incorporated into an eDNA survey? I love this idea. For eDNA's potential limitations, one of its really amazing strengths is just how easy the environmental samples are to take. There's lab work here, the pipetting on a lab bench, there's some finesse on that side, but in the field, eDNA is so easy. I've done eDNA sampling with elementary school students. We've seen no contamination in our field blanks from doing eDNA with school groups. And there's a number of ways in freshwater that you could take these eDNA samples. You can mail syringe samplers with screw-on filter capsules where someone can take a water sample, push it through the capsule, store it in a tube of silica desiccant gel beads, mail it at room temperature, and get DNA detections back at the lab. So I think there's huge potential for schools and community volunteers to take eDNA samples across the landscape in something like, stream watch or riverkeeper programs or lake associations. I think the challenge with that is when I think about really successful kind of citizen science programs, and I think about, I don't know, something like eBird, right? There's this incentive, people love birding, and if you can record data while birding, people can use that data, but the person collecting the data is getting a lot of reward out of, well, I like birding anyways, I want to go do this. Taking eDNA samples is not very interesting. And there's often a time lag before you get your answer. And I think one of the things we'll need to do to make eDNA really viable for community environmental monitoring programs or citizen science is to work on kind of the turnaround time and the presentation of data back to data collectors, where if it's a school group that's doing a stream watch program, has combined maybe invertebrate sampling with taking a water sample for eDNA, can we get them, a result, maybe whole community meta barcoding? Here's the fish you found in your water sample. Can we return that to that classroom in 2 weeks instead of 3 months because there was a bottleneck at the sequencing center on campus and we couldn't get the samples run, right? I think time to get samples back to kind of community environmental monitors will make it more possible to unlock maybe these kind of diffuse volunteer monitoring of the environment with eDNA, which I think has enormous potential. Now I've fallen way behind. Yeah, a question about conventional methods and eDNA and cost effectiveness. We have not studied it enough. There have been a few good studies that have compared eDNA to conventional sampling on cost. Nate Evans has done some good ones for common fisheries methods against eDNA. We have one in prep for alligator snapping turtles in Louisiana on Department of Defense lands, where they're very hard to detect by trapping. And we've compared the cost of our trap net nights for alligator snapping turtle against eDNA surveillance for snapping turtle. And this paper is kind of in development, but it's something like a degree of magnitude less money to have run eDNA for alligator snapping turtle, just because the personnel time, the hours to do the trap set nights are so expensive. I always want to kind of caveat those cases where eDNA is cheaper, that there's also a cost there, that there's data eDNA can't collect. Our Boston Mountains crayfish, where our conventional sampling doesn't do much better than occupancy, eDNA is pretty comparable. But if we collect an alligator snapping turtle in a net, you've got a length and a mass and blood work and physiology, and that stuff may be worth that effort. I've got a question. I really liked hearing about the alligator snapping turtle study. I've been thinking about the different kinds of research experimental opportunities associated with reintroductions, and this was a great example. Yeah, I loved the alligator snapping turtle study. Seemed so exciting to me to have very high confidence in where the organism we were looking for was, and not in an enclosure or a stream cage, but living how an alligator snapping turtle wants to live. When we started the study, I said, oh, this is going to be perfect. You know, Ethan, there's all the turtles have radio tags on them and we know where they are. Halfway through our eDNA sample collecting, Ethan would go scuba dive or snorkel to recapture his turtles and take measurements and get mass. And Ethan's much braver than I am, because I wouldn't scuba dive under undercut banks just trying to hand collect alligator snapping turtles. Ethan came up from one of these undercut banks with a turtle, and they got it up on the shore, and it was not one of theirs. It was not a tracked turtle. It was an actual wild turtle that had dispersed into Illinois, and it was the first, you know, non-reintroduced alligator snapping turtle in the state in something like 80 years. And I thought, oh wow, well, we know where most of the alligator snapping turtles in Illinois are, but a little of that noise is, yeah, maybe there's a few out there that didn't grow up in the Peoria Zoo. Peter asks, most of the work you discussed was based on invertebrates and benthic ones. The biology and physiology of fish is very different. Mucus, large, yeah. How might release mechanisms and fates of eDNA by these two groups of organisms differ? I will say, you know, those studies trying to relate biomass or abundance to eDNA concentration. I recently peer-reviewed a follow-up meta-analysis on that analysis that disaggregated the studies into fish versus crayfish versus freshwater mussels. And they found stronger relationships under field conditions between fish than crayfish or mussels. And you know, I think it may be true that we can get closer to good biomass or abundance estimates for fish just from the biology of the organism than these benthic invertebrates. And you know, that meta-analysis has small sample sizes on the benthic invertebrate size. It's a handful of crayfish studies, it's a handful of mussel studies, but it feels supported to me by people like Mike Spear, who just finished a PhD at the University of Wisconsin, and he's a postdoc now at the Illinois Natural History Survey. They did work relating, you know, walleye kind of stock assessments from Wisconsin DNR to eDNA concentrations in Wisconsin lakes, and they also had pretty good performance for walleye. I mentioned in the talk that I started as an eDNA skeptic. I work on crayfish. The first eDNA study I did was with a student volunteer at Notre Dame who wanted to do the work attached to some of our routine crayfish monitoring. And I said, yeah, sure, but it'll never work. It's a crayfish. It's in an exoskeleton. It's in the bottom of the lake. When it worked really well, I kind of had to readjust my outlook on eDNA, but it worked well for occupancy. It didn't work well for relative abundance. And that may be a persistent difference between invertebrates and fish.
Dr. Larson, if you want to just take this one as a last question,
Yeah.
that would be awesome.
One more, and I'll let people go. Similar to this question, do you think lower DNA shedding is a possible explanation of why you need so many samples to get a detection of the black warrior water dog? Yeah, you know, I know there's interest in doing laboratory studies to kind of quantify shedding rates between different organisms to kind of inform what DNA production should look like in the field. I think that's potentially valuable if we find that we can kind of generalize DNA production across taxa generally, that if you're this type of salamander, this is how you shed, how much DNA you shed relative to a crayfish does, you know, it's 10 grams of crayfish produce less DNA than 10 grams of live fish in an aquarium. You know, there may be some generalities we can get to there. I worry that if that's not true, we have so many, let's say, threatened and endangered species that we're unlikely to have those lab studies on, that I wonder if we won't kind of collect that variable detection probability better from field studies than lab shedding studies. But it may be that we do get to a meta-analysis that says freshwater mussels, regardless of species, shed this much less eDNA than a crayfish, which sheds this much less than a benthic fish, which sheds this much less than a water column fish. That may be someplace where we are in 5 years or so. I think our black warrior water dog, it's just very rare. You know, we have similar organisms like hellbender salamanders that are also very rare, that eDNA works pretty well for, but I think the water dogs ESA listing was sadly very warranted. It's a very tough animal to find.
Hey, thank you, Dr. Larson. We appreciate you coming and taking the time out of your day to give this presentation. It was awesome. For those of you still on the call, we have Julia Parrish from University of Washington next week, talking about some citizen scientist stuff. So if you're interested in that, tune in again. And yeah, we'll see you guys next week. Thanks again, Dr. Larson.
Yeah, thanks for having me.